Can see your mouse.
Great. Thank you so much, Katty. And thank you so much for inviting me and for organizing this talk. So I'm excited to share with you some of my work done on the diversity and origins of prokaryotic viruses on sinking particles reaching the abyssal ocean. So my name is Elaine Lewell, and I'm a postdoc scholar and microbial oceanographer at the Woodsville Institution. And some of this work was done during my PhD at the University of Hawaii. So I'm interested in viruses because they're the most abundant biological entities in the ocean. So if we were to take a drop of seawater in the surface, we find that the number of virus-like particles are on average an order of magnitude more abundant than other life forms such as cells. And so in particular, double-stranded DNA bacteriophages, which are viruses that infect bacteria, are abundant in the open oceans that cover roughly 40% of our planet. So one way that researchers can study these life forms is through microscopy. And so here I'm showing you 3 different physical types of viruses that's common in the open ocean. And what's conserved about all of them is that they contain DNA inside a protein capsid. And this genetic diversity represents a reservoir of largely unexplored diversity in the ocean. So I'm interested in studying these microbial communities on sinking particles because it connects the surface and deep ocean. So you might have heard of this process called the biological carbon pump. And this is a process in which primary producers in the upper ocean, so typically in the upper 200 meters, fix carbon into their biomass. And this carbon is then aggregates into sinking particles, and that reaches the deep sea. And so a lot of things can happen to these sinking particles as they reach the export depth of 200 meters, and they reach the sequestration depth of 100 meters in this twilight zone here. And one of these things that could happen is that microbes can colonize these particles and start decomposing it. And so this transforms carbon from particles into carbon dioxide and decreases export efficiency. So what we know from previous studies is that these particles harbor diverse prokaryotic communities, including microbial decomposers. And we also know from microscopy that they harbor virus-like particles. What we don't know is which viruses live on sinking particles. So what are their diversity and who are they infecting? Where do they come from? Were they sort of colonizing the particles as they sink, or were they transported from the surface into the deep sea and potentially play a role in export processes? And what's their correlation with carbon flux? If we can identify populations that are, for example, positively correlated with carbon flux, then these populations could play a role in mediating export. And so there's 2 conceptual models as to how viruses might impact export processes. One view is that viral lysis decreases export by releasing cellular carbon into dissolved organic matter. So this dissolved organic matter can then be respired by heterotrophic bacteria in the upper ocean. And this shunts carbon away from export into a respiration, being recycled in the upper ocean instead of being exported to the deep. And so another view is that viruses could increase export by enhancing aggregation and sinking. And this view is supported by experiments done on large eukaryotic phytoplankton. So the open oceans are characterized by smaller cells. So I was really curious how these dynamics might play out there. So Station Aloha is an open ocean environment in the North Pacific subtropical gyre, roughly 100 kilometers north of the Hawaiian Islands. And it's the sampling site of the Hawaii Ocean Time Series. So we have access to over 30 years of environmental data through depth and time to provide us some context to our work. So we know that the surface waters is characterized by high productivity and low nutrients. And this in part drives the small cells that dominate this sort of environment. And the abundant primary producers include Prochlorococcus, a small cyanobacteria, as well as some Synechococcus. And the abundant heterotrophs include SAR11, so a very small and ubiquitous cell, as well as some known particle degraders. So the deep chlorophyll maximum occurs at a depth of around 125 meters. And below this, we see a marked increase in standing stock of nutrients due to a decrease in primary productivity in the mesopelagic. And so we've studied some of the microbial communities along this water column. So we know some of the viruses that are abundant here, as well as their cellular hosts. And so I'm curious to also study viruses on particles in this environment to link the surface ocean to the deep. And to do this, I analyzed a 3-year metagenomic time series of sinking particles collected at 4,000 meters at Station Aloha. So we're looking at microbial communities that likely traveled all the way from the surface to the deep and were sequestered to the deep sea. And these samples were collected on bottles that rotate every 10 to 14 days. So we're looking at both time and depth integrated data. And these 21 samples span most of the year. And so this trap is set out yearly and it's taken out. So here I'm showing you the sampling points along with the particulate carbon flux. And what we see here is that some years have this feature called the summer export pulse of heightened export. And so we can look at which viruses positively correlated with carbon flux to identify ones that might be implicated in export processes. We can also look for overlaps between these 4,000 meter viruses and viruses that came from the surface to identify which ones were transported from the surface to the deep. And so what I found is multiple lines of evidence that's consistent with the idea that viral lysis could enhance export. So from these deep sea viruses, I assembled roughly 800 viral population genomes. So you can think of this as a sort of a species of viruses. And some of them, 260, were also found in shallow water samples in the upper 500 meters. A subset of these viruses were also found on sinking particles from out of the euphotic zone. So these were collected at 150 meters. So what we're seeing here is planktonic viruses that are found on particles sinking out of the euphotic zone and into 4,000 meter particle traps. And so this is strong evidence for vertical transport of these viruses. And so then I was curious who these viruses are, or what are some of their properties. And so I've identified these populations by whether they positively correlated with carbon flux or not, and by their reproductive strategy. So whether they were these lytic active type of viruses that were likely infecting and killing their hosts, or whether they were passive. And so what we see is a subset of these vertically transported viruses that also positively correlated with carbon flux and appear to be actively killing their hosts. And so this is consistent with the idea that viral lysis could enhance export. So then I was curious who these players might be. Who are they infecting? And so we're just sort of starting to uncover viral diversity in the ocean, particularly in the deep sea. They're novel. And so to identify them without reference databases, I looked for clues from their cellular hosts. And so my colleague Andy Liu, now at the University of Queensland, generated metagenome assembled genomes. And in these genomes, these cellular genomes, we can look for these passive viral sequences called a prophage. And so we can look for matches between the deep sea virus with these prophage regions to link it to its host. We can also look for CRISPR spacers. So some of you might have heard of CRISPR, this gene editing tool. And the CRISPR spacers basically serve as a record of previous viral infections. So sometimes when a virus infects its host, and for some reason it's not successful in killing the host, Sometimes the host might take a piece of that viral DNA and retain it in the genome to remember that virus for later. And this is how the technology of CRISPR is based on that ability to recognize a specific sequence and cut a new virus that comes in to prevent it from infecting their host. So by using these approaches, we can we can then identify viruses to their host. And I found that viruses that correlated with carbon flux were largely linked to heterotrophic hosts. So here I'm showing you the abundance of some viruses that positively correlated with carbon flux. So for example, the summer export pulse event. And we see that they target bacteria that are known to consume organic matter in particles. So for example, vibrios are a known organic matter degrader. And so these results sort of pose a new perspective as to how viruses could mediate export process, in that if active viral infections prevent microbial decomposition of particles, then this could increase export efficiency. So I found that some of these viruses that were exported from the upper ocean to the deep sea were linked to small primary producers. So so here I'm showing you the abundances of viruses that were vertically transported from the upper 500 meters into 4,000 meter particles traps. And of those that I was able to identify, they either targeted cyanobacteria, so these small primary producers, or bacteria associated with cyanobacterial blooms. So this work raises a question of whether viruses of small primary producers could enhance export through lysis, aggregation, and sinking. And so this work was recently published, and you're welcome to check out the paper for more details. And to summarize, we found that viruses throughout the upper 500 meters were exported to the deep sea on sinking particles. Some of these viruses positively correlated with carbon flux, including those that are known to target organic matter degraders. Some of these vertically transported viruses targeted small primary producers. And so this work highlights the need to consider the functional roles of viruses to predict their impacts on carbon cycling. So we can use metagenomic approaches to identify which hosts that they're targeting, because depending on who they target, they can have very different effects on export processes. For example, viruses that target organic matter degraders might prevent particle degradation and enhance export. And viruses that target small primary producers might increase aggregation and sinking and enhance export. And so the overarching goal here is to model microbial impacts on the carbon cycle and to integrate these in situ metagenomic approaches with mechanistic models to help us understand export processes that's happening across our oceans. So I want to wrap up by inviting you to come work with me. I'm starting a lab next year, so feel free to reach out at any point if you're interested in viruses, metagenomics, and marine microbial ecology. And with that, I thank you very much for inviting me, and I welcome any questions.
Great, thank you so much, Elaine. That was super interesting. Does anyone have any questions before we move on? Well, I have a bit of a question. So yeah, it's a lot to unpack here. I'm really interested in reading your paper now. Have you considered a temporal component to kind of what you're seeing? So is there a change in community composition in different seasons or over time?
Yeah, that's a great question. So what I found is that there is super high variability in terms of microbial assemblages on these particles, in particular for the viruses. and that these patterns are not reproducible between year to year, even during periods of high summer expert pulse. So for example, some of the viruses that popped up during 2015, we don't see them again the next year in the summer export pulse. And this is super curious because the planktonic assemblages in the upper ocean are fairly consistent between seasons and also between years. And so I think what is showing here is that these particles are just highly heterogeneous and diverse. They can come from multiple sources. There's probably some sort of spatial and temporal patchiness as to which particle just happens to land in the sediment trap as well. Yeah, that's a great question. We should have a lot more sediment traps in the ocean.
Exciting. Yeah, that is. I would like them too. Yeah. Oh, right. Yeah. Oh, well, thank you so much. Does anyone else have any questions?
Hello.
Yep.
Can you hear me? I have a question, Dr. Wong.
Hi.
Hi. Thanks for the talk first. Then my question is that from your talk, that is a lot of viral particles are shifting from top to the bottom. So that may increase the biodiversity in deep sea zones. But my question is there is some way to shift from these particles, this virus from bottom up? No.
Oh, that's a great question. I mean, I feel like, so Amala has a question and she's a physical oceanographer at Hui. I feel like the physical oceanographers here might have something to chime in. Yeah, I imagine how things might look differently at upwelling zones, for example, on the East Coast, on the West Coast of the US, as well as South America. But yeah, I wonder if this is a great question for physical oceanographers. And I'm curious if Amal could chime in.
Well, instead of the vertical transmission by water current, is there some animals shift from, daily shift of microbes from bottom to up?
Right. So from what I understand about copepods or that do these dieo vertical migrations, it seems that they graze near the surface and then they go to the bottom. And so if I'm envisioning this, I can sort of imagine vertical transmission the other way from top to bottom as well, if they're sort of pooping deeper than they're eating. But yeah, I wonder if there might be animals that graze in the deep and then they come up to the surface.
Okay. Thanks for the question. Yes.
Yeah, thank you so much.
Yeah, hi Elaine. I have a question too. So what is the fate of the products of lysis? So when they lyse a cell, like what happens? Is it like, would you consider dissolved organic matter? Is it labile or is it refractory, or what ends up, or what happens to all that organic matter?
Yeah, that's a really great question. I imagine that's fairly labile, simply because that cell was recently living. And so I imagine it's fairly juicy for heterotrophs to come in and take advantage of that. So depending on the size of the lyse products, I imagine that I can end up both in dissolved and particulate pools. So larger pieces of cellular debris likely become POM, and smaller amino acids and pieces of proteins likely become in the DOM pool. Yeah, it's a great question.
Yeah, thank you. And just one other question is, do we think that there are more viruses in the surface ocean or deep ocean, or deeper, or mesopelagic, right? So do we know that, or?
Yeah, that's a great question. So from counts, and this is, I assume, largely planktonic, so it's not really accounting for particles. There is 1 or 2 orders of magnitudes of decrease as you go from the surface to roughly 500 meters. And from what I know of fairly or even deeper environments, such as hydrothermal vents, or the areas surrounding hydrothermal vent, that number drops further. And so we can imagine something similar to the Martin curve of particles, where we see an exponential decrease of biomass with depth. Hi, Clarissa.
Hey.
Oh, thank you, Anwar. Hey, nice to see you.
I have a very basic question. I was just wondering what drives that the viruses get into the particles. So are there certain factors that make it easier for them, or are there certain sources of viruses that end up in the particle?
Right. Yeah, that's a great question. So the way that I think about it is this could happen either during formation or so that's actively during formation, or passively through absorption. So for example, if a virus is actively involved in killing a cell, and when the cell is bursting or sort of aggregating together to form the particles, we just see some viruses being trapped in that organic matrix. That I imagine that's how it can get trapped. And another way is through absorption. So we know that some viruses just irreversibly stick to organic matter. And that's another way in which they could passively contribute to export as well.
Thanks.
Thanks, Clarissa. Hi, Yang.
Hi. Hi. Is that me? Yeah, cool. Yeah, I'll ask my question. Great talk. That's really exciting data. So I may have missed it, but have you shown the suspended particle data? I think HOT also has suspended. If the virus, as you said, is passive, so supposedly there should be more virus in the suspended particle data. Have you seen the similar signals in your bottle particle data, for example? Do you have the insight on that?
Have I seen similar signals in what particle
The
data?
bottle particle, nesting bottle. I mean, right now you're showing sediment traps, right? I'm just curious. That's sinking particles. So what's going on in the suspended particles? Have you seen higher viral abundance, etc., those kind of things?
Right. Yeah. So so I did some work on. On sort of particles along the upper ocean. Viral particles, but this I really mean sort of the free viruses that are planktonic and floating around. So, and from this work, both of the viruses and cellular hosts, we know that in general, the suspended or planktonic fraction is a lot more diverse than the sinking one. And so I think what is happening is that there's probably a lot of viruses just floating around and coexisting in this environment. And a subset of them, a subset of their cells are really good at growing on sinking particles. And that tends to be, particle degraders. And so that's why we see a sort of a overrepresentation of a specific type of both viruses and cells on sinking particles. So it's sort of a selection process, who decides to colonize and do well on sinking particles.
Oh, that's great. That's interesting. So I'm not really a biologist. I'm trying to wrap up. So let's say suspended particles and sinking particle, initially, they have maybe possibly the same composition of virus, viral-like composition. However, because sinking particles is more juicy, so it's preferentially going to select particle degraders, let's say. Because if you imagine sinking particles is from aggregation of suspended particles, supposedly virus is going to have the same composition, but there's a selection process. Because does that make sense? Is that correct?
Yeah, that's what I imagine is happening, because particle degraders can reproduce much faster than planktonic bacteria. And yeah, it is a very juicy source of carbon, particularly in these low nutrient oceans.
Cool. Thanks so much.
Thank you for the question.
All right, great. If there's no more questions, we might transition to Xingwei. Elaine, thank you so much for sharing with us. Really interesting talk, and look forward to chatting with you in the future.